Around 6.5 to 7.5 million years ago, our evolutionary path split from that of our closest relatives, the ancestors of the chimpanzees, marking the beginning of a journey that would lead to the emergence of the modern-day human. Since then, the human and chimpanzee genomes have undergone various transformations, leading to the vast phenotypic differences between the two species today. We may share some similarities, but overall there is quite a striking dissimilarity between us and them. It may be surprising then to learn that the human and chimpanzee genomes are over 98% identical at protein coding loci. How can a genome be so similar and phenotypes be so different? It is hypothesized that gene regulation may be the answer here. Researchers from the Georgia Institute of Technology have determined that sequences outside of the protein coding genes within the genome of each species contain a large amount of variation. Specifically, large genomic gaps that exist in one species but not the other. These gaps are collectively referred to as indels, or insertions and deletions, and their absence or existence can affect how a gene is regulated and expressed. Researchers began categorizing and comparing the indel variation within or around genes between humans and chimpanzees using bioinformatic tools such as Ensemble. Interestingly, researchers found that 70% of the indels categorized were homologous to sequences of retrotransposons that integrated into the human genome following the divergence of the two species, the majority being long and short interspersed elements. Researchers then used microarrays containing samples from the brain, testes, heart, liver, and kidney to compare gene expression levels in the different tissues from each species. They found there was a significant correlation between sequence differences and expression differences between species. This research is significant in furthering our understanding of human regulatory evolution and the mechanisms behind it, and it illuminates the potential importance of retrotransposon insertion in the human genome as a positive driving force of our evolution. My learning from GEN2052 allowed me to understand the mechanisms by which retrotransposons work, and how this could contribute to indel variation through their ability to jump from one location to another throughout the genome. I could also comprehend how regions in close proximity to protein coding genes are crucial for gene regulation, as they contain numerous regulatory elements. Other areas of learning which allowed me to comprehend this article included bioinformatics tools like Ensemble, which allow the comparison of human and chimpanzee genomes, and the use of microarrays to measure gene expression. However, an area of the research that presents limitation lies in correlating genetic differences between species to the level of gene expression. This is because genes are expressed differently at different life stages and in different tissues, and therefore just because a significant correlation between genetic difference and gene expression level isn't present in one life stage and in the tissue of one sample, doesn't mean that at another life stage and in a different tissue a significant correlation won't exist. I also found myself asking how differing levels of expression of the same gene can be correlated with differing biological function that cause such dramatic phenotypic differences between the two species. My knowledge of transposable elements was cloudy, an abstract concept that consisted of useless DNA arbitrarily jumping around the genome. However, they fascinated me, and this article helped reinforce the mechanisms through which transposable elements operate. I wanted to explore the potential functions of transposable elements, seeing as they make up such a prominent part of our genome. This article has left me with many new questions. To what extent do transposable elements influence gene regulation? Could we further quantify their activity to deepen our understanding of evolutionary mechanisms and how humans may continue to evolve? How much have retrotransposons contributed to the significant divergence of the human genome from our ancestors?